of a capture-recapture experiment, but in brief, just in case you're not. So for a capture-recapture experiment, the study will involve going out to a study area, collecting some individuals and marking them in some way so that they're individually identifiable. And then you'll re-release them back into the population. And on subsequent occasions, you'll go out and try and capture individuals, some of whom will be new, some of whom will be previously captured. So essentially, the type of data that we get from capture-recapture data is a matrix of zeros and ones, where the row of the matrix corresponds to an individual and the column of a matrix corresponds to the capture occasion. So that matrix will literally just have binary information contained in it. For the way that we're going to present this model, it's sometimes useful to be able to pick out each row of that encounter matrix that we have. So we use the notation that HI corresponds to the encounter history of individual I. So that will be that we're simply taking the row I of the full encounter history matrix. And some useful values that we can calculate from that particular encounter history for individual I is that we can record what the first capture occasion is for individual I, which we denote by FI. So essentially that will be the first one in that row vector, the time of the first one, and the last capture occasion, which I'm denoting by delta I, which will be the final time the individual is seen. For the purposes of the species that we're going to be looking at and thinking more broadly in for translocations, we're going to assume that new individuals might only start entering the population at a lagged time after the beginning of the study. So we're going to use TOR to denote the occasion that new arrivals start entering the population. And I'll discuss that a little bit more with the case study. And then simply capital D is going to be the number of observed individuals. So what we're really interested in is the underlying population size. And over the course of the study, we observe capital D individuals, which will, of course, be an underestimate of the total population. I should say, if anyone's got any questions as we go along, please do feel free to interrupt me. OK, so that's the data. We've got some nice capture-recapture data. And the Jolly-Sieber model is a capture-recapture model that's been around since the 1960s. It's kind of well established. And the reason we're interested in this particular model of capture-recapture data is that it allows populations to be open. So it allows for new arrivals into the populations. It allows for deaths from the populations. So you're able to estimate demographic parameters of survival and arrival probabilities, as well as being able to estimate population size. So we use the notation that we have a phi to denote the survival probability, P, which is our nuisance parameter, which is essentially the detection probability or capture probability, and the betas represent the arrival probabilities into the area. So as I mentioned before, we're going to be assuming that new arrivals don't arrive necessarily from time one. And so we have this period where the betas, these proportions of the populations that arrive, are actually fixed to 0 for the time before TOR. And because these betas correspond to proportions, the sum of the betas has to equal 1. So we have those constraints on the parameters. Now, for the model itself, the parameter we'll actually estimate will be capital N. So capital N is sometimes referred to as the superpopulation. And the superpopulation is basically defined to be the number of individuals who have ever been available for detection at some point during the sampling period. So it's basically, if you sort of take every single individual who's kind of been in and out of your study area, how many of them are there? But what actually interests us for the purpose of our study in terms of conservation translocations is actually this derived parameter of the number of individuals who are going to be there at time t+1. And you can see that we can express that in terms of the parameters that we've estimated from the model. Okay, so the Jolly-Sieber likelihood itself is essentially a multinomial likelihood. So you can think about constructing the probability of each of your observed encounter histories. So we have the probability of the HI here for all observed individuals. So it goes from I equals 1 up to D. We've got our multinomial coefficient term, n factorial over n minus d factorial. And then we've got this additional term, P of H0. Now, what H0 corresponds to is if you think about what would be the encounter history of an individual that you haven't managed to capture. So that encounter history would basically be a string of zeros. And so within our population, we've observed capital D individuals, but in total, there are going to be capital N individuals. And so there are going to be N minus D individuals who have that 0 encounter history. So that's where that form of the likelihood is coming from. And I'm not going to go into kind of great detail about these equations, but essentially it's possible to write down the probabilities of these observed encounter histories, HI, and also the probability of, I want to say the probability of observing the unobserved individuals. So the probability of basically a string of zeros. So that's the probability of the H naughts. And we can write those down in terms of those data values that I've spoken about. So the time of first capture or the time of last capture, the Fi and the delta I, and then in terms of the parameters of our model. So it's a really nice model. It's also a great source of projects for MSc and undergraduate students as well. So there's lots of really nice examples of applications of these models, and there's various R packages that you can use to fit the standard Jolly-Sieber model. Okay, so to begin with, when we were thinking about, well, how do we analyze this data from a translocation, our default reaction was, okay, well, we'll just use the standard Jolly-Sieber model and we'll impose some parameter constraints on that model. Now, theoretically, that should be possible. So this is a translocated population. So the difference between the translocated population and a normal population for a Jolly-Sieber model is that we know that the total number of individuals who are there at the start of the study is just equal to the number we have put there. So we know that value of N1 in a translocated population. And the problem is, when trying to fit these Jolly-Sieber models, say using some existing R software, it's not possible to fix that initial population size. So we try to do it indirectly. We've tried to fix the probability of capture at time 1 to be equal to 1. And we tried to put constraints on the betas to make sure that things were fixed. But essentially, all of the R packages, and there's a number of different R packages that you can use to fit the Jolly-Sieber model, essentially we encountered problems with all of them, and none of them would give us sort of sensible estimates. So what we decided to do instead, because we were hitting this barrier of just using the existing model, we thought, okay, well, let's try and produce a likelihood that really directly models what we have in the data set. So what we decided to do was that we wanted to account for the translocated individuals in the population. And when we do that, what we want to do is have a term which accounts for the probability of the translocated individuals, but we condition on the time at which they're released into the population. So for anybody who's familiar with the capture-recapture literature, it's a little bit like having a component of a model that's a Cormack Jolly-Sieber model, and then another component which is the Jolly-Sieber model. So the Cormack Jolly-Sieber model basically doesn't concern itself over estimation of population size. You just say, okay, well, I condition on the fact that I've captured X individuals at time 1, and then I'm going to estimate my survival probability and capture probability. So by having this component for the translocated individuals, this doesn't muddy the water when it comes to estimating actual population size later on. So essentially, the adapted likelihood that we've got for these translocated populations, it looks very similar to the Jolly-Sieber model we had before. But we've now just got this additional term, which corresponds to the probability of observing those translocated individuals. So we assume that there are N0 translocated individuals, and the total population size is still equal to N. But for individuals who are born into the system, so not those that are translocated, we assume that there are D minus N0 wild-born individuals. So in total, we've got D observed individuals, but N0 of those correspond to translocated ones. Okay, so that's the kind of the likelihood, and it all looks messy. We actually ended up coding this both in R and MATLAB. So we found MATLAB a lot more efficient for the optimization. So it was just an ease of trying to get lots of simulations and lots of bootstraps and things for confidence intervals. We have got R code that works. It's just a little bit slower. So the first case study corresponds to a lesser night gecko. So I don't know if anyone's familiar with these. I certainly wasn't before the study, but it's a small nocturnal gecko that's endemic to Mauritius. So you can see it's really small in size. This is somebody's fingertip. And these gecko are only found on the 4 outer islands surrounding Mauritius. And they're currently classified as vulnerable by the IUCN. Now, lesser night gecko are individually identifiable by their pattern on their back. And so they take photographs to perform the capture-recapture study, and then those are run through a program called Hotspotter to be able to construct the individual encounter histories. Now, the aim of this whole study is to re-establish a population of lesser night gecko on Round Island. So the source population is going to come from Gunner's Coin, which is this island that's shown in the top picture, and Round Island is very aptly named, photo from the bottom. So there's a number of considerations that we've got to take here. So we've first of all got to think about what's the impact of removing the lesser night gecko from Gunner's Coin. It's a relatively small population. If we remove 80 to 90 individuals, what impact is that going to have? We also want to think about what's the potential impact on the native species on Round Island. So Round Island has a number of species which would be predators to lesser night gecko. So in order to think about that, Katie's been thinking about different release strategies to optimize the chances of survival of the lesser night gecko. Okay, so for any of you that don't know the geography of Mauritius, so you can see that Gunner's Coin and Round Island are off the north coast of Mauritius. And Katie's PhD started in October 2019, so right before COVID. The plan was originally that she'd be going out to do fieldwork in 2020 to prepare for this translocation, but obviously we were all desk-based. But thankfully, there had been a previous translocation of lesser night gecko in some of these southeast islets off the coast of Mauritius. So in 2011, 30 lesser night gecko had been moved from Île aux Vacois to Île Marianne. And so we had a nice translocation data set that we could really use to kind of explore some of our new statistical methods with. So I'm just going to show you kind of one output from the modelling that we've done. So this is from the historical translocation, that one from Île aux Vacois to Île Marianne. And the translocation happened in 2011. And the population has been sort of studied, it's been sampled for at least once a year since then, not always at the same time of year. So we do have to account for kind of differences in time interval. But on this plot, the darker line shows the estimates of population size over time, and the dark bands correspond to bootstrap 95% confidence intervals around those estimates. What's shown in the light grey was the best that we could do trying to use existing Jolly-Sieber models through R packages. So we were trying to kind of artificially fix population size at the beginning. So there's just a couple of things to note. So clearly this translocation has been a success, that the population has established itself. What we should note, though, is that the population remains very small until about 2015, and then the population growth starts to happen. So that's I'm told that's a very sort of common feature when populations are establishing themselves. Really importantly, so when the managers are going to be asking us, has this translocation been a success? It's going to be the estimates at the start of this period that are really important. So you can see that in 2011, we've got no error around our estimate because we know how many geckos were translocated. And you can also see that we've got quite tight confidence bands on those estimates of population size, which is really going to help reassure the managers that we can get quite precise estimates. And that really comes from the sort of appropriate constraints that we're putting on the system that we're modelling. Okay, so just to mention that Katie has now done the other translocation. So just before Christmas, she went back out to Mauritius and has translocated the gecko from Gunner's Coin to Round Island. I think Katie's PhD has been kind of full of challenges, because another thing that hit this study area was that there was a massive oil spill off the coast of Mauritius in late 2020. And so although the main aim of her PhD was really to kind of perform this translocation and to sort of work out whether the population has established, she's also been looking at aspects of what impact has the oil spill had on the reptiles as well. So it's proved to be a really interesting, if diverse, project, and quite different from the one we intended to. But the capture-recapture data is now being collected post-translocation. So we're looking forward to seeing how the new methods have done. There's also, I mentioned that during the translocation, it's been important to think about different release strategies. So following sort of expert elicitation, we worked out that we needed fencing areas around the areas where the gecko are establishing themselves, and then those fences would be removed. And a cyclone decided that those were going to be removed slightly earlier than we had originally planned. So the cyclone brought those down a couple of weeks ago. So it'll be interesting to see what happens in terms of the dispersal of the gecko as well. So that's kind of work in progress, but hopefully a future story to tell. Okay, so the second case study, it also comes from Mauritius. So Telfair skinks are one of the largest skinks in Mauritius. So we're kind of going from one extreme to another in terms of sizes. But basically, the Telfair skink went through a period of really rapid decline in the 16th century. And it was thought that the decline was due to the arrival of people who were taking boats over to the islands. And so what happened, a number of invasive species arrived on the island, so goats, rabbits, and rats. And you might be thinking, well, hang on, how are goats and rabbits actually going to affect skinks? But I'm reliably told that the herbivorous invasive species kind of degrade the habitat that the Telfairs actually relied on. So they relied on that habitat both for food and shelter. And as the rabbits and goats sort of destroyed that, then the population of Telfair declined. So the species actually became extinct in most of the archipelago, with the exception of one location, and that was Round Island. So there have been many conservation efforts, mainly run by Mauritian Wildlife Foundation over the last 30 years. And one of those was to remove those invasive species, so eradicating rats and rabbits on Gunner's Coin. And then back in the early 2000s, there were sort of lots of ecological assessments of Gunner's Coin to work out whether it would be suited for the reintroduction of Telfairs. And at that point, 250 Telfairs were translocated from Round Island back to Gunner's Coin. So I really like this system because there's a kind of two-way movement of some of these species. So previously, Gunner's Coin had been the source population for lesser night gecko, whereas now it's the sort of host area for the translocation. So one thing to be aware of during translocations is you need to think about genetic diversity of the species and the population. So following the translocation in 2007, there was a further reintroduction of 100 additional skinks in 2012. And the telfairs, they're not individually identifiable, but they were fitted with a microchip, which allowed for individual recognition. So during this study period as well, this translocated population was also then used as a source population for further conservation translocations. So this is a kind of, it's a data set that represents a kind of mixture of, it was a translocated population to begin with, but then became a source population, which has made it a really interesting case study for what we're interested in. So we've fitted, or I should say Faye has fitted numerous models. This is just a snapshot of the top ranked models. We've considered lots of different parameter structures within this, so time dependence, sex dependence, accounting for sampling effort, so how many nights sampling had been taking place. I mentioned there's an uneven time interval, so we'd looked at accounting for the uneven time interval, but allowing survival to be constant and things like that. And we also included potential effects for, are the translocated individuals, do they have a lower or higher survival or capture compared to wild-born individuals? So after lots of model fitting, lots of waiting patiently, we basically have ended up with a model where time dependence is important for the arrival of new individuals. We've got sex and time effects in capture probability, and we've got an additive sex and time effect in survival probability. And it was one of the first applications I've worked on where we've kind of got a nice clear gap of AIC. Normally I'm used to kind of models that have a difference of 2 decimal points or something. So that was nice. So we've got estimates of population size for both the males and the females. So you can see by the spacing of these estimates that unequal time period that I was mentioning in terms of the sampling. So the green vertical lines represent where additional translocations happened. So the nice thing about the model structure that we've presented in terms of that adapted Jolly-Sieber model, it easily accounts for periods where you've got kind of multiple translocation periods. Whereas if you were just trying to do it constraining a Jolly-Sieber model, that would be really challenging. The red lines correspond to points at which removals happened. And unfortunately, we don't know which individuals were removed. So we've had to account for these individuals within the calculation of the total population. So the difference between the estimate in 2014 and the estimate in 2015 has taken those removals into account, but we haven't been able to extract specific individuals from the analysis. And then the picture for female telfairs looks very similar. That if I was criticising my undergraduate students for their graph plotting, we should perhaps put these on the same scale. The female population is noticeably smaller than the male population. Okay, so that kind of covers the aspect of how do we model the translocated individuals. Now I want to kind of take a step back and think about the source population. So we want to think about what is the effect of conservation translocations on that source population. And considerable work has been done about removal or depletion models. So I got involved with doing some removal modelling specifically for great crested newts. So great crested newts are a big thing in Kent because they're sort of a protected species by the UK government. And if you find great crested newts on areas of land that you want to develop for property, there needs to be appropriate measures taken to ensure that the great crested newts are removed from the area prior to development. I think those rules have now been slightly relaxed, but previously they had to provide evidence that they had cleared the site of great crested newts. So removal data essentially just corresponds to counts of individuals. So how many individuals have you removed on each sampling occasion? And if you assume closure within the population, essentially you can model this using a nice straightforward geometric model. And you can think about it being, well, the probability that I capture individual at time t, I will have failed to capture it t-1 times, and then I finally captured it. But that's a very sort of simplistic model, and there are many extensions that exist. So Eleni and I did some work expanding this model to allow for populations to be open. And then one of our PhD students who was analysing some removal data of common lizards adapted the model to allow for temporary emigration. And the reason for that, the common lizards bury underground for periods of time and then come back up above ground. So you needed to account for the fact that they became temporarily undetectable while they were underground. So if we want to assess the impact of the source population, we really need to think about, well, obtaining those robust estimates of N, as I mentioned, but we need to do that while accounting for the removal of individuals. And there are a number of problems with this. So as I mentioned before, detection probability can be quite low. Removals for translocations are typically done over a small number of sampling periods. So for the lesser night gecko, they were all done over 3 days. And it can be really challenging to estimate N just from removal data, especially if the number of removal periods is small. So we were sort of set the target of thinking, well, is there any way that we can get improved estimates of N while doing the removals? And given that we'd been working with the capture-recapture data for the translocation, we then thought, okay, well, could we supplement the removal data with a short period of capture-recapture prior to doing the removals? So I apologise for my diagram here. I tend to think of things visually, and I know not everyone does, but I'll try and explain what my diagram's showing. So the red period here, the red section corresponds to the period of capture-recapture. And within your total population N, you'll have some individuals who are caught during the capture-recapture setting, which are shown in dark red, and you'll have some individuals who are missed during the capture-recapture study, and they're shown in the pale red. And then the blue period shows the subsequent removals that were happening. So once again, the dark blue corresponds to individuals who were removed, and the light blue corresponds to individuals who were not removed. So the reason we're looking at it in these 4 different scenarios was because when we were thinking about constructing the model corresponding to this, we wanted to make sure we were accounting for all of these possible combinations. So essentially, you could have both being dark red and dark blue. So that would correspond to individuals who appeared in the capture-recapture data and who were removed. But then you might have some individuals who appeared in the capture-recapture data who were not subsequently removed, and that corresponds to the dark red and the pale blue. And then when I was presenting the initial Jolly-Sieber model, I mentioned the individuals who are not captured at all. To this bottom block where you've got pale red and pale blue. So those are the individuals who were not caught in the capture-recapture and not caught in removals either. So you can see that we've got capital D observed individuals, and that corresponds to some who are in capture-recapture and removals, some who are just in capture-recapture, and some who are just in removals. Okay, so the actual structure of the model is going to look fairly similar. So this sort of builds on the idea of the Jolly-Sieber model that I've explained, but we're just accounting for the fact some individuals are removed. So obviously, once they've been removed, essentially the remaining part of the probability of their observed encounter history just needs to fall away because they've been removed, so you don't need to continue modelling the probability they've survived, etc. I should apologise, I'll just pop back. The tour here, I realised when I was putting together these slides, this tour as tour in the previous part of the talk. So sorry, because I brought together two two different projects, I've managed to muck up my notation. So TOR here corresponds to the number of sampling occasions for the capture-recapture part of the study. Okay, so I'm not going to go into details because I don't think anyone wants complex statistics like formulae explained to you on a Wednesday afternoon. But essentially, the likelihood function is once again a multinomial in structure. So we've got parts which correspond to the capture-recapture, we've got parts that correspond to the removal. And the eagle-eyed amongst you might notice that this first square bracket, the content inside that product from j equals 1 up to TOR, and the content from j equals TOR plus 1 up to Li is actually the same. And so really we could combine this just to be from j equals 1 up to Li. But the issue is that when we remove an individual, so if we have the last sighting being less than TOR plus 1, we need to make sure that we don't have any subsequent contribution within the likelihood term. So we've presented it in this way because we need to have that clause when we program it. So yeah, it's possible to construct this joint likelihood. So we looked at various simulations, sort of how advantageous is it having these periods of capture probability. And really we're focused on settings where we have small capture probability because that reflects what we'd have with these reptiles. So here we're looking at varying the amount of capture-recapture data, so varying that TOR. Starting with no capture-recapture data and moving up to having all 6 encounter occasions being capture-recapture data. So here we're in a setting where population size is very small, so we've only got 100 individuals in total. But what you'll note is I mentioned that it's very difficult to estimate population size from removal data alone. And that's evidenced here by the really poor estimate of detection probability when you just have removal data. So the true value is 0.05, which is all the way down here, and we're estimating it to be 0.3. You can see things get marginally better when you've got one period of capture-recapture and the rest removals, but really you need to get up to 2 periods of capture-recapture or more to be able to get sensible estimates of P. Now, obviously, P is a nuisance parameter, but it has knock-on effects to the estimation of population size. So you can see that the overestimate of detection probability, capture probability, corresponds to an underestimate of population size. So we can see the kind of inverse relationship in these box plots of population size. I should say that once you increase N, once you increase your population size, this picture doesn't look quite as stark. So if you've got a reasonable number of individuals, even with a low detection probability, you do start to get semi-sensible estimates. In fact, I did add them in. So that this is the corresponding picture when you've got 500 individuals. So this is the estimate of detection probability and the corresponding estimate of population size. Okay, so we thought, okay, well, that's great. But as I mentioned with the telfairs, we don't know which individuals were removed. And it's more likely that you're not going to know which individuals are removed, because typically the capture-recapture data that you'll have from your source population might well be collected separately from the captures which are going to be done for the removal and translocation. So going back to my diagram, essentially what that means is that, yes, we've got some capture-recapture data that's represented by the red, and yes, we've got some removal data that's represented by the blue, but now we know the number of individuals that have been removed, which we're going to denote by D star, and we know the number of individuals that have been caught on the capture-recapture experiment, which I denote by little d, but we no longer know capital D. So we no longer know the total number of individuals that have been observed during the study. And the reason for that is that we don't know this estimate of pale blue in here. So we kind of know parts of it. We know that capital D must be at least as big as the maximum of D and D star, but we don't know any more than that. Now, there's a couple of ways of dealing with this. So one is that it builds on some ideas that I did. I worked on a lot during my PhD and subsequent fellowship, that we can construct something called an integrated model. So we could basically say, okay, well, we've got some capture-recapture data, we've got some removal data. Let's just assume those two data sets are independent. We can construct a likelihood for the capture-recapture, we can construct a likelihood for the removals, and then just multiply them together. Now, obviously, these data sets are not independent. So we wanted to kind of investigate, well, what are the potential problems of assuming that they are? So exactly as I've just said, we can construct a likelihood for capture-recapture, which I'm going to denote by LCR here, and we can write down the likelihood for the removal, so that kind of geometric model that I described. And we can construct this integrated likelihood, which is simply the product of the capture-recapture likelihood and the removal likelihood. So what we wanted to do, we kind of knew what the real was, because we had the likelihood that corresponds to the joint data, which corresponds to, you know, when you know which individuals are which. So that's really the truth. And then we've got this integrated likelihood. So what we then looked at was algebraically, how do these two different likelihoods actually differ? So what we're able to do, we're able to write the integrated likelihood as a product of these two multinomial terms. So you can see that the n over n minus d star, that comes from the removals. So the d star corresponds to the number of observed removals, and little d comes from the number of observed in the capture-recapture experiment. And then I've got a term capital lambda here, which just represents the rest of the algebra. And what we can show is that actually the true likelihood, so that joint data likelihood, looks very similar in structure. So we have the capital lambda. It only really differs in terms of this multinomial coefficient. So in the integrated approach, we have these two terms in terms of d star and d. In the truth, we have just n factorial over n minus d factorial. And if you remember, that capital D corresponds to the total number of observed individuals. And that's the detail that we're missing when we don't have that information of which individuals were removed. So we were quite excited by this. We thought, okay, well, surely what we need to do then, we need to come up with another way of approximating that likelihood. So instead of having those two multinomial coefficients in the integrated likelihood, let's instead have an approximate integrated likelihood where we have some kind of d tilde. And the d tilde will be an estimate of the number of observed individuals, taking into account both the capture-recapture and the removal data. And we explored a number of possible ways of approaching this. So we thought, to begin with, well, can we just actually directly estimate d tilde? Could we have it as a parameter in our model? We looked at using the EM algorithm, replacing that d tilde with the expected value of d, and the expected value of d can be written in terms of parameters we are estimating, so in terms of the superpopulation and the detection probability. And both of those approaches failed, because I think the model that we're trying to fit is actually parameter redundant. So what we ended up finding was that d tilde was always converging to the boundary of the parameter space. So it would always converge to either d or d star. So we were rather disappointed, because we really thought, okay, we've got a neat trick here. We could have done a better job. What we've ended up settling for, and it is very much settling for, so if anyone's got any bright ideas of how to tackle this, I'd be really thrilled to hear them. We've gone for a Horvitz-Thompson-like approach. So we're going to estimate the other model parameters, so the p in this case, or in the open case p and phi, and then we use that to estimate n. So having done all of that, we were then thinking, okay, well, we've got to compare three different things here. We know We've got the joint model, so when we know the individuals that overlap between the data sets. So that's represented on the first box plot. We've got our integrated approach, where we're assuming independence between data sets that are actually dependent. And then we've got our approximate D-bar approach, which we're going to use using the Horvitz-Thompson approach. And I have to say, we were quite disappointed because we went to all the effort of trying to correct our our integrated likelihood, and essentially it hasn't really helped. We may as well just assume the two data sets are independent and multiply those likelihoods. So what is shown here, and it's quite striking, is that if you know the individual identities of the individuals that you remove from the population, you can really get better estimates of your superpopulation. So that's definitely a kind of a major take home for our ecological partners in this, that when they're doing these removals from these islands in particular, it's worth running the IDs of those individuals through a system to make sure that we can account for properly removing them. So when I presented this work previously, it was picked up, there's a slight bias in that box plot in the middle, the integrated model. And that is something we will look at slightly more closely, but I'm not sure whether it's going to be a kind of major issue with these types of approaches. And when we increase population size, you can see even more that our D-bar approach and the integrated approach, it really doesn't have very much difference at all. But once again, the joint truth does come out to be important. Okay, so there's lots of ongoing work. So as I said, this is very much work in progress. We've developed the equivalent joint models for open populations. So the ones I presented on the slide are for closed populations, where you have no new arrivals, no departures. We're going to be looking at applying this methodology to the Telfair case study, so to account for those removals that happened partway through the study. And what we then need to think about is how can we think about improving the forecasting approaches for removals from source populations. So really providing a tool for people conducting the conservation translocation planning to give them some reassurance that taking individuals from a source population is not going to adversely affect the underlying population there. Okay, so there's a few references. So one of those papers, the top one, which is on the modified Jolly-Sieber model, that's currently under revision. So if anyone would like a copy of it, I'd be very happy to share it with you.